So tonight's video is going to focus on DNA structure, a little bit of RNA review in there. We'll talk more about RNA when we get to transcription and translation, as well as DNA replication. So first up, let's talk a little bit about the purpose of DNA. So the purpose of DNA is the fact that it's our primary source of heritable information or genetic information that we're going to pass on. Now, in some cases, RNA can be as well, but for most organisms, it's DNA. But again, we need to kind of keep that in the back of our minds that RNA can do these things as well if need be. So DNA is going to both store and transmit our genetic information. So it'll store the genetic information within the organism, and then it will transmit it to the offspring, and it will transmit it to the next generation of organisms. And again, RNA can do this as well. Prokaryotic organisms, so when we talk about DNA chromosomes, prokaryotic organisms usually have their chromosomes in a circular form. If you look up here on the top right, you will see that their chromosome in that prokaryote is circular. Where in eukaryotic organisms, they usually have multiple linear chromosomes. Okay? Yes, the DNA is wound into the double helix, but again, we're talking the overall shape of the chromosome. We're not talking about the double helix part. So for instance, in this picture down at the bottom, this would be a prokaryote. There's no nucleus, there's no membrane-bound organelles. And if I took that red DNA out, it would be shaped like a circle. So prokaryotes have circular chromosomes, or eukaryotic organisms have usually multiple, and they're going to be linear chromosomes. However, both kinds of organisms can contain what are called plasmids. Plasmids are little small extra chromosomal, so they are not part of the main chromosome. You'll see how these plasmids here, all three of them, they are separate and they are not incorporated into the bacterial DNA. So they are what are considered extra chromosomal. And so they are these little extra chromosomal pieces of DNA. They are going to be circular, whether they're in a eukaryotic or a prokaryotic cell, doesn't matter. The plasmid will be circular and they will contain particular genes. And we'll look at some biotechnology type experiments that can manipulate these plasmids and how we can use those plasmids to transfer information between organisms to induce variety, to help synthesize proteins, just a variety of uses. So we'll talk about those a little bit more when we get some further into biotechnology. So in reviewing the structure of DNA, remember DNA is going to be double-stranded. RNA will be single-stranded. Like I said, most organisms are using DNA to pass on their genetic information, but some organisms will use RNA. So DNA is double-stranded, whereas RNA would be single-stranded. Remember, DNA is made of a nucleotide. Nucleotides consist of the sugar, phosphate, and the nitrogen base. Right? Our sugar for DNA is deoxyribose, where our sugar for RNA is just ribose. So DNA contains our deoxyribose and RNA contains ribose sugar. They both contain nitrogen bases. Okay? Remember our base pairing rules, right? We have A to T and C to G. However, in RNA, right, it's A to U. U takes the place of T. U does not pair with T. It takes the place of T. And these base pairing rules are conserved throughout evolution. So what this means, this shows common ancestry. It shows relationships. And it means that organisms millions of years ago, adenine went with thymine and cytosine went with guanine. So this process has been conserved as evolution has occurred. Okay, these base pairs are held together by hydrogen bonds. If we look over here at the picture on the far right, we can see our hydrogen bonds holding these base pairs together. Remember, hydrogen bonds are relatively weak bonds. They're pretty easy to break, and that's going to be important because we're going to need to break this apart to be able to replicate DNA. So these weaker hydrogen bonds are holding these base pairs together. So we've got our double-stranded DNA made up of lots of nucleotides, right? Like we can see a nucleotide over here. I've got the phosphate highlighted in yellow there, the sugar, deoxyribose, which is in blue there, and then I've got a nitrogen base, in this case guanine, in the orange. And if you will notice, some of the nitrogen bases have 2 rings, like this adenine down here at the bottom. This green adenine has 2 rings, and the guanine has 2 rings. The ones with 2 rings, a double ring structure, those are called purines. So my purines, my spelling is over here on the left. My purines consist of adenine and guanine, and they have a double ring structure. So my pyrimidines are this, you can see my thymine, my cytosine, how they have a single ring structure. Uracil in RNA will have a single ring structure. Okay, y'all need to remember too, like A is for adenine, G is for guanine, C is for cytosine. If all you've ever known is A, G, C, T, and U, make sure you get their actual names down. Okay, so my pyrimidines are a single ring structure, and that's my cytosine, uracil, and thymine. And you'll notice as you look at the pairing, I always have a pyrimidine, so 1 ring, with a purine, with a 2-ring structure. So 2 pyrimidines, C and T, can never go together. 2 purines, the A and the G, can never go together, right? A purine goes with a pyrimidine, in particular A to T, C to G. A saying that can help you remember this is that my pyrimidines are cut. Okay, and if you stop that right there, pyrimidines are cut. Cytosine, uracil, thymine, those are my pyrimidines. Pyrimidines are cut from purines. While this is not a true statement, okay, I did not take an adenine here, right, that has its 2-ring structure, cut it, and now I have a thymine. Okay, that is not what happened. However, okay, to help you remember it, my pyrimidines are cut from purines. That means my purines have to be the bigger molecule. They have to be the bigger. They have to be the bigger nitrogen base. They have to have the 2 rings if I'm going to cut something from them. Okay, I also always just kind of think of the big word. The bigger the word, the smaller the molecule. So the bigger the word, the only 1 ring. The smaller the word, I've got my double, my 2 rings. The pyrimidines are cut from purines. So these structural characteristics, in particular my base pairing rules, my A always going with T and C always going with G, these are what allow DNA to be used as hereditary material. And RNA has those same base pairing rules, just A to U instead of A to T and C to G. So again, these are what the basis of what allows DNA, these nucleic acids, to be my genetic material. So DNA replication is important because it provides continuity of life. Okay? By passing on this DNA, I'm passing on my heritable information. And by passing on the heritable information, I create continuity within the stream of life. Okay? And so the way DNA is replicated helps ensure that this hereditary information, okay, stays consistent. Okay? DNA replication, if y'all remember, is considered semi-conservative. Okay? Semi-conservative. So by semi-conservative, we mean that I am going to use 1 strand as a template for my new complementary strand. So if I'm looking at this picture here, you can see my original DNA is this blue. And so I have 1 original strand, 1 original strand. They're both being used as a template for my new strands that will be built using my complementary base pairing rules. And so when I'm all done and I get this strand of DNA and this double strand of DNA, half of it is the original strand and half of it is new. So that's where the semi-conservative part comes from. Okay? The fact that as the DNA is copied, it uses 1 of the original strands to act as a template for adding a new strand to it. So when I'm replicating my DNA, I've got a wide variety of enzymes that are going to come into play. And so we're going to go through and we're going to look at a number of these. So the first one that we're going to start with is helicase. Please do not write down any and all of these enzymes just start blindly writing them. You only need the ones that we are going to talk about. Okay, so starting here with helicase. Remember, ASE is our enzyme. Helicase unwinds the DNA. So it opens up the DNA at what's called the replication fork. My replication fork here is where my 2 original strands of DNA, you can see them branching off. So it's where my 2 original strands of DNA branching here, that's my replication fork. And helicase is unwinding that DNA. So basically what helicase is doing is it is breaking the hydrogen bonds between those nitrogen bases. So in front of that helicase, you have what's called Topo Isomerase. That one is quite the name to spell there. So Topo Isomerase. Topo Isomerase prevents what's called supercoiling. Basically, if you think about this DNA, if you think about a piece of yarn, and we'll kind of talk about this in class, right? If you pull apart the ends of a piece of yarn, it will start to get really, really tight in front of where you're pulling it apart. Okay? Sometimes it'll even kind of knot up in front of it here, because as I pull those 2 ends apart, you'll see it spins, it spins, it spins, it spins, it spins. And so Topo Isomerase prevents that from tearing and getting knotted up in what is, like I said, what is called supercoiling. Okay, it's going to be unwinding the strands a little bit to release the stress. So it releases the stress that the helicase unwinding puts on the front end of that DNA. Okay, so now that we've opened up the DNA, we're going to have some various enzymes that are going to actually lay down the new parts. So we have, first of all, we're going to have DNA polymerase. Okay, DNA polymerase, again, ACE, an enzyme. I'm going to be laying down, DNA polymerase is going to do a couple of jobs. And we have multiple kinds of DNA polymerase, and we're going to talk just about 2 of them. So we're going to talk about DNA polymerase 3. You'll notice right here it says contains POL123, that's Roman numeral 3 there. So DNA polymerase 3 is important for the synthesis of DNA. However, for DNA polymerase 3 to work, it requires RNA primers. So you can see on your picture here, you've got this enzyme, this big yellow enzyme in the middle here, RNA primase. Okay, so it's going to lay down an RNA primer. You'll see how there's these little green parts on the front of the new DNA that's formed on this bottom strand. The RNA primer would be out here. It's already gone. Okay, so the RNA primer has to be laid down for DNA polymerase to then come do its job. You can see this. So I've got these green RNA primers here, and then I have this DNA polymerase, this purple one, moving in this direction, laying down new DNA. So DNA polymerase 3 is important for DNA synthesis. It requires these RNA primers to work. So once the RNA primer is down, then that lets the DNA polymerase know that it can continue to lay down new DNA nucleotides. So that's what the DNA polymerase is doing, is it's laying down new DNA nucleotides. And we're going to look at a picture, a different picture of this in a second, so that if you're not getting it from this picture, you might get it from the next picture. So it's laying down these DNA nucleotides. And you'll notice in this picture that you start to get, how the 2 strands look a little bit different. How I've got one strand that is nice and long and continuous, the one on the bottom, and I've got one strand that is broken up into these little fragments or these little pieces. So let's talk about that for a second. So DNA polymerase 3 can only lay down in what's called the 3' direction. If you think about other pictures of DNA that you've seen, what will happen, DNA is anti-parallel. If you remember that from your freshman year, the fact that I'm going to have these opposite ends. So if I have a strand of DNA here, I'm going to have a 3' end at one and a 5' at the other. Oops, that's a 5. And then the opposing strand is going to go the opposite direction. If y'all remember either laying down your gloves or your notebooks in class, and they had to go opposite directions, that's that anti-parallelness. And the 3' and the 5' end are going to be referring to the way where the phosphate group is versus the sugar. That part isn't really important for y'all's purposes. Okay, so what you need to realize, though, is that DNA polymerase can only lay down nucleotides in the 3' direction. So on this little example strand over here, on the right, it can go down, but on the left, it goes back up like that. So it can only lay down in the 3' direction. So what that means is that for this bottom strand, my 3' direction is near the replication fork. So it's just continuously laying down. No big deal. My 5' is at this end, which means this is my 3'. This would be my 5' end of my original strand. So on the top, right, if I look at my original strand, on the bottom I have the 3' end, which means this is my 5' end on the top here, which means on my new strand, my 3' end is going away from the replication fork. So that means I have to synthesize my DNA away from the replication fork. Well, I can't do that in one continuous process because the helicase doesn't unzip the entire DNA. So it unzips a little. We lay a little piece of DNA down in that direction. It unzips some more. We lay this next little piece. It unzips some more, and we lay this next little piece, working in that 3' direction. So what I get is what's called a lagging strand versus a leading strand. So the lagging strand is the one that's going to be synthesized in pieces or fragments. They are also called Okazaki fragments, named for the person that discovered those. So, oops, I spelled that wrong. Okazaki fragments. So those are found on my lagging strand. It's lagging because it's a little bit behind, right? Because it's got to go a little bit at a time, open some more, a little bit at a time, open some more, a little bit at a time. And we're going to model this in class too, and so I think that might make more sense. My leading strand is one continuous fragment all the way. So it doesn't, that isn't an issue. I don't have, I'm not going to have these little pieces or these little fragments. So my DNA polymerase 3 is laying down new nucleotides in the 3' direction. So it's continuous. So I get my one RNA primer that was way out here where it first started. And then I have all these little RNA primers every time I need to start a new fragment on my lagging strand. So I have another enzyme, DNA polymerase 1. So DNA polymerase 1 is going to replace these RNA primers with DNA. I can't have little hunks of RNA left over in my new strand of DNA. So I have DNA polymerase 3, which builds based off of the primer, then builds DNA, my new DNA. DNA polymerase 1 removes the RNA primer and replaces them with DNA nucleotides. And then also on that lagging strand, I'm going to have this enzyme ligase. Okay, so DNA ligase is going to ligase or stitch my little strands together, my Okazaki fragments together. So I'd have DNA ligase come stitch those together, come stitch those together. So in the end, I end up with one big, long, continuous strand that's all DNA, because DNA polymerase 1 came in and replaced the primers, and then DNA ligase joined it all together. Okay, so let's look at a different picture of this, and we'll kind of talk through it again real quick. Okay, so again, I've got all my enzymes here. So in this case here, the green one is my helicase. So it's unwinding the DNA. So in front of it, I would have that Topo Isomerase, making sure that the DNA didn't get damaged as it gets unwound. So there's the helicase breaking the bonds between. On this time, on the bottom is where I have my lagging strand. My strand that's going to be in little fragments. I can see my DNA ligase here, putting the fragments back together. Remember, this is being synthesized in this direction because it can only add to the 3' end. So I have my RNA primers right here that are being laid down. DNA polymerase 3 is adding the new nucleotides. DNA polymerase 1 is replacing those RNA primers with DNA nucleotides. and then the DNA ligase is stitching all those fragments together. We're on the top, on that leading strand. That means this is my 3' end. Okay, I'm just continuously adding DNA nucleotides using DNA polymerase 3 just straight into the replication fork, so that I will end up with 2 new strands, okay, that are, again, semi-conservative, half old DNA, half new DNA. Like I said, we're going to model this in class, and I think that will help a lot. Your goal right now is you need to make sure you have all these enzymes down so that you can be prepared to use them as where you can have your notes and you can use them as you're doing the modeling. And you know what each enzyme does, and you can just continually practice with them.